Let me start. So first of all, thank you very much for giving me an opportunity at this wonderful ICMRBS ECR webinar series. And today, the title of today's presentation is the oligomeric assembly regulating mitochondrial HDRA2 function as examined by methyltrosy NMR. So human high temperature requirement A2 HDRA2 protease is a mitochondrial protease which mainly resides in an intermembrane space of mitochondria. The main function of HDRA2 is to cleave unfolded and misfolded proteins and protect cells from these unfolded protein stresses. In the right panel, the structure of HDRA2 is shown. Each subunit is composed of a pink protease domain followed by a PDZ domain, and the crystal structure was solved as a 100 kilodalton homotrimer. In this slide, the structure of monomeric subunit is close up. The catalytic center at the protease domain is formed by these aspartate, histidine, and serine residues, and this catalytic triad is covered by this blue PDZ domain in the crystal structure. Therefore, this structure is thought to represent an inactive conformation. Other biochemical studies have shown that the hydrophobic activator sequence, such as GQYYFP, can bind to this blue PDZ domain and then allosterically activates the protease domain. However, the structures of the activated conformation or the substrate bound state is not available. Therefore, the structural mechanism underlying this allosteric communication has remained unclear. Another important feature of this HDRA protease family is that this protease usually functions as an oligomer. For example, in the case of bacterial DECP protease, DECP is known to form a wide range of oligomer from trimer to 20-former. However, in the case of human HDRA2, only a trimeric structure has been reported, and the formation of higher oligomer is not reported in the literature. So the question is, can HDRA2 form a higher order oligomer? And if so, what is the physiological oligomeric state of this protease? So the research questions are as follows. What is the physiological oligomerization state of HDRA2? And how does the substrate bind to the PDZ domain? And how does the PDZ domain allosterically regulate the proteolytic activity? First of all, I'd like to start with a characterization of the oligomeric state of HDRA2. As the molecular weight of HDRA2 protease is about 100 kilodalton as a trimer, we utilized methyltrosy technique, which selectively observed side chain methyl signals. In the NMR analysis, we used S306A mutant in which the catalytic serine is replaced with alanine to suppress self-cleavage. Here, the methyltrosy spectra of HDRA2 is shown, and we assigned all of the isoleucine, leucine, valine, and methionine methyl signals by combining NOE analysis and mutagenesis approach. During this assignment process, we noticed that some methyl-containing residue gave rise to two different NMR signals, and the relative intensity ratio of the two signals changed as a function of total protein concentration, pointing to the fact that this protease is in an equilibrium between different oligomeric states. Here I show two examples, valine 396 and valine 452. And at high concentration, we mainly observed H state or high molecular weight state, while at the lower concentration, we mainly observed this L or low molecular weight state. We could also find an I41V mutant here, in which only the L state signal is observed. To rigorously assign these oligomeric states, we analyzed small angle X-ray light scattering and calculated the molecular masses of these proteins. The molecular mass of wild-type protein was in between trimer and hexamer, while the molecular mass of this I441V mutant was consistent with the trimer. From these results, we concluded that H and L state represent hexamer and trimer respectively. We also analyzed this oligomerization equilibrium by NMR. In the left panel, we calculate the affinity for hexamerization by dilution experiment and calculate the signal intensity of hexamer and trimer at various protein concentrations. the affinity was on the order of 1 to 10 micromolar Kd, depending on the salt concentration. And in the right panel, we measure the interconversion rate of these oligomeric states, and we could find that the exchange rate was about 20 per second at 40 degrees by ZZ exchange experiment. This result was quite intriguing for us because the formation of this hexameric state was not reported in the literature. So we sought to characterize the structure of this unprecedented hexameric state by using NMR. First, we analyzed the chemical shift differences between hexamer and trimer. In the left panel, I show the overlay of the HMQC spectra obtained at high and low protein concentrations and calculate the chemical shift differences between these two spectra. And here, as shown here, I show the plot of the chemical shift differences, and we map the methyl residues whose chemical shift differences were large onto the structure. Significant chemical shift perturbations between hexamer and trimer were observed at the C-terminus region of this blue PDZ domain, suggesting that this PDZ domain mediates the hexamerization interaction. Then we model the hexameric structure by rigid body docking. To get the overall structural information, we analyzed the tax profiles obtained at 4 different protein concentrations. The hexameric HRA2 is a barrel-like structure stabilized by intertrimer PDZ-PDZ interaction, and the structure was consistent with the results of CSV and mutation analysis. Then the question is, what is the physiological role of this hexameric state? To address this question, we analyze the binding affinity of the activator peptide and compare the affinity between hexamer and trimer states. For this purpose, we used this short hydrophobic activator peptide, DDGQYYFE, which can bind to the PDZ domain and then activates proteolytic activity. We confirmed the binding of this peptide by using NMR, and interestingly, very similar chemical shift differences between free and bound and full length and the isolated protease domain were observed. This result suggests that the peptide bound conformation is very similar to the isolated protease domain. This result suggests that the tightly packed PDZ domain is dissociated from the protease domain upon the binding of the peptide. Then we analyze the binding affinity of this activator peptide by analyzing the titration experiment. Here I show 3 representative methyl signals, and in most signals, we could observe a single apparent free state signal and single apparent bound state signal. However, in the case of the leucine 377, we could observe both hexamer and trimer signals in the apo state, and the population of hexamer was larger than the trimer. However, in the presence of a small amount of peptide, we could observe the population of the trimer state was larger than the hexamer. And we could also observe a single apparent bound state signal. This result suggests that the binding of the activator peptide accompanies the population shift in different oligomeric states, and the binding affinity toward hexamer and trimer are very different. To make the long story short, I analyzed this titration profile based on this thermodynamic model. In this model, the peptide does not bind to the hexameric state and only binds to the trimer state. And the binding of this peptide occurs in a stepwise manner, and then the fully bound trimer further oligomerizes to form a bound hexamer. This thermodynamic model can be described by using these 5 association constants, and we fit the NMR signal intensities to obtain these 5 association constants. However, the problem was we could not resolve all of these states in the NMR spectra. So we had to interpret the signal intensities by using a different combination of concentration terms. For example, in the free state signal, this free state signal is actually an overlay of free hexamer, free trimer, 1 bound trimer, and 2 bound trimer, and all of the bound protomers in each particle contribute to the free signal intensity. And the scaling factor 36321 means the number of the free protomer in each particle. In the same way, this bound signal intensity can be represented as an overlay of 1-bound, 2-bound, 3-bound trimer, and a fully bound hexamer. And bound protomers in these states all contribute to the bound signal intensity. In the case of Lucene 377, we could resolve the hexamer and trimer state in the free state signal, and the free state signal is again an overlay of 3 trimer, 1-bound trimer, and 2-bound trimer. And the bound signal is again an overlay of 1-bound, 2-bound, 3-bound trimer, and a fully bound hexamer. In this way, we could convert the concentration terms into the NMR signal intensities, and we could fit the NMR signal intensity at various activator peptide concentration and could get 5 association constants. These slides show the graphical summary of the thermodynamic model along with the obtained association constants, 5 association constants. The interesting thing was the second binding affinity was tighter than the first, and the third binding affinity was tighter than the second. So that means there is positive cooperativity in the stepwise binding reaction. So in summary, we found that HRA2 is actually in an oligomeric equilibrium between hexamer and trimer state in solution environment. And this hexameric state is a self-protected state in which the peptide cannot bind. And the activator peptide can bind to the trimeric state in a stepwise manner. And in the binding reactions, there is a positive cooperativity. And the bound conformation can be characterized by an open conformation in which the PDC domain is dissociated from the protease domain. And this presumably explains the higher catalytic activity in the bound conformation because now the catalytic center is exposed to the solvent. So we propose that HRA2 activity is dually regulated via oligomerization and the formation of tight interdomain interface and preventing it from non-specific binding of substrates. Finally, I'd like to thank my supervisor, Luis, and my colleague, Rolf, for helping me conduct this research. And I'd like to thank the funding supports for this research. That's it. Thank you for listening.
Thank you very much. That was great. So we're going to pass straight on to our next speaker, Nicola.
Yeah. All right. Can you see my slides?
Yes, it's good.
Okay, good. So thank you very much for this opportunity to speak today and to present our work on intrinsically disordered proteins. And in particular, oops, come forward. Okay, yes. So as most of you probably know, a significant percentage of the proteome of eukaryotic cells is significantly disordered. And this means that we have to use an ensemble of diverse conformations to represent their functional state. Now, this gives an advantage to IDPs, the fact that they can access unique binding modes that are not accessible to folded proteins. And all these binding modes rely on the dynamics of interconversion between states in the ensemble. So this is what we want to understand. And to do so, because we participate in this seminar series, the best experimental technique is by far NMR, because IDPs give very beautiful spectra, very sharp lines, in which each peak reports on ensemble divergence information. And in particular, if you are interested in picosecond to nanosecond dynamic, what you want to measure is spin relaxation. And in particular, we measure spin relaxation as a function of two properties. First, as normally done, we measure at different magnetic fields. So the idea is that by measuring at increasing fields, we can scan the spectral density function as much as possible. But we also change another variable, which is temperature, because by changing the temperature, we actually change the shape of the spectral density function. And from the temperature dependence of the points that are probed by spin relaxation, we can say something about the physical origin of motions. So in practice, this is an example of a data set measured on our favorite model system, the C-terminal domain of Sendai virus or entail. So in this case, we measured about 62 relaxation rates at 4 different temperatures and 4 magnetic fields. And we and other groups, for instance, at Palmer's lab, we realized that we need 3 components to fit all this data. So 3 different time scales, fast, intermediate, and slow. There is no temperature dependence of the fast time scale. And we can model all these 62 data sets in one go, simply by assuming a pseudo-Arrhenius relationship for the intermediate and slow time scale. And in this way, we can extract some pseudo-activation energy of these modes. And this is what actually tells us, gives us insight into the physical origin of these dynamics. Now, long story short, we can assign these fast modes to local librations, intermediate dynamics to the sampling of backbone dihedral angles, and slow dynamics to solvent-dependent motions that are segmental in their nature. And this is, now I don't have time to go into all the analysis that we did, but one simple example that should convince you that this slow motion actually are of chain-like nature is that if you actually repeat this study with construct of different lengths, so green, blue, and red from long to short, you see that the only parameter in our analysis that actually changed is the time scale of these slow motions. Now, if you want to understand a little bit better what's going on, it is very convenient to complement your NMR analysis with MD simulations, because using MD simulations, you can calculate rotational correlation functions for your NH bond vector that you can and then you can use a Fourier transform to convert this information into NMR relaxation data. And these, at the same time, while you are looking at your copy of the protein, exploring the free energy landscape. So you really have a very good idea of the physical mechanism behind these dynamics. Now, doing simulations of IDPs is actually difficult. Now, during the years, we came out with an ensemble selection in the time domain procedure, in which basically what we do is to carry out an ensemble of conformations, and then we use a subset of our data to select a sub-ensemble of trajectories that best represent the information that we have on dynamics in our data. Now, we can perhaps discuss this in the breakout room. What I want to tell you now is that if you apply this scheme and you actually characterize these segmental dynamics in IDPs using different force field and different water models, you realize that slow dynamics actually depends very much on the properties of your water model. And in particular, it correlates very well with the lifetime of hydrogen bond and the temperature dependence of this lifetime. Now, this opens a lot of interesting perspective because, as you know, the hydrogen bonding properties of water is tuned by things like osmolytes and micromolecules. And you have to remember that we want to actually understand what is the significance of all these dynamics in a complex environment, such as the cellular environment where these IDPs work. So the idea is, the question is, can we actually develop a model that accounts for this complex environment? And the answer is actually yes. And I have to derive this model, we started by measuring relaxation as a function of the concentration of viscogen. Now you see that in general, if you increase the crowding, for instance, R2 goes up, and so do the heteronuclear NOE. And we, at the same time, we also started measuring water R1, because this is actually a measure of the rotational correlation time of water molecules, which is a probe of viscosity. We can then define this parameter rho, which essentially measures how much friction changes with the amount of viscogen that we are adding to our buffer. And if we do so, we realize that the timescales of our dynamic mimic the dependence of friction, of solvent on friction, but this dependence is about 3 times as big for slow dynamics at intermediate, reflecting the fact that slow dynamics is reporting on more long-range processes. Now, you can actually then derive an analytical formula that takes into account both the dependence on the amount of crowding and the temperature dependence. So from this analysis, on top of the parameters that we extracted before, we also derive this friction coefficient epsilon, which are site-specific for intermediate and slow motions. And this is what we did here. And this results in a very robust and portable model that we cross-validated by applying to a completely different IDP, MKK4, and also by using a completely different Crowder, PEG, instead of Dextron as the previous slide. And actually, this can be exported to any kind of medium, as complex as you like, provided that you can actually measure R1 of water that allows you to define this parameter rho. And this is what we did here going into cells. So we measured the relaxation of MKK4 in oocytes. And you see that using our model, we can actually reproduce very well and actually predict very well how this the parameters measured inside. So to conclude, I showed you that temperature-dependent spin relaxation provides insight into the physical origin of motions in IDP, and that we can also use MD simulation to understand a little bit better these motions, and in that NMR relaxation is highly dependent on the environment, and a lot of this dependence can be understood by measuring relaxation as a function of viscogen. And that's all. Just would like to thank Martin for his guidance in this work, and two PhD students with whom I had the pleasure to work with, Anton and Victor, and all of you for your attention.
Okay, thank you very much indeed. Okay, so we're going to go straight on to our final speaker, Enrico.
Yes, so thank you also for inviting me to participate in this nice webinar series. So I will show you some of our recent results that we obtained by applying in-cell NMR approaches to study protein-drug interactions within human cells, both in a static way and in a time-resolved way. So since quite a few years, I've been working in Florence at CERM with Lucia Banci to develop and apply in-cell NMR methods. So for the, I guess many of the audience are familiar with in-cell NMR, but I will shortly summarize why one would ever want. Nicola already mentioned the importance of the intracellular environment. NMR is really powerful because it provides a direct way to get information at the residue level or even at the atomic level on structure and dynamics of biological macromolecules directly in the cellular environment. So it's a very powerful tool in that sense. And it can also therefore obtain functional insights on proteins, and as I'll show you, also provide information on the interaction of macromolecules with ligands, which is, I think, of great importance in the field of drug development. So how do we do in-cell NMR in human cells? So we have, a few years ago, we have developed a method for overexpressing proteins in mammalian cells. We have adapted an existing protocol, and this is very suitable for in-cell NMR because it allows reaching quite high expression levels, and we can perform isotope labeling in human cells. This is one more methodology which is complementary, I would say, to the other methods to deliver proteins inside human cells, which rely on delivery by cell-penetrating peptides or toxins or electroporating the cells themselves to introduce proteins. So with any of these methods, you can observe intracellular proteins, labeled proteins, and get structural and conformational information. So we sought to understand whether this method could really help in the drug development workflows, because there is a very, as many of you know, a very high attrition rate when one does rational drug design, starting from the in vitro studies, structural studies, or activity studies, when you screen thousands of compounds and then you select the most active compounds. And then at some point, you go towards more in vivo assays, cellular assays, and then finally some animal models. And at that point, most of these drugs will just fail for a series of reasons, which very often are due to the fact that within the cells and then tissues, the drugs don't do what they're supposed to. Although the binding affinity and inhibition could be very beautiful in vitro. So in cell NMR, I think it's very suited to help in this process, somewhere between the lead optimization in vitro and the in vivo assays, so preclinical assays. And to prove these, to assess this capability, we started working with human carbonic anhydrase 2, which is a very beautiful model target, which is also pharmacologically relevant because the isoform 2 is one of 16 isoforms of CAs, which are several which are involved in a series of pathologies in humans. So they are actively targets towards which there is a very active development of potential drugs. So by expressing this protein, we assessed the behavior of the protein. The protein is well folded in cells and is free to tumble. So the R2 are not really affected by interactions inside the cellular environment, which means we can get very nice HSTC spectra, and which also means that we can immediately monitor ligand binding inside the cell. So we started treating the cells with a series of compounds, some of which are actually approved drug, like acetazolamide and methazolamide as control compounds. These are sulfonamides, which bind as a fourth ligand to the zinc active site of the protein. And in cell spectrum, we are in a bit a lucky case, I should say, because we have a few proton signals which fall in a region of the spectrum, which is typically the immuno region, where there is basically no background interference from protons from the rest of the cell, which means that we can even avoid recording 2D spectra and go for a more efficient screening by 1D NMR. So this allowed us to assess the binding of a series of compounds which were developed, a few reference ones, and then some other compounds developed by Claudio Sucura group here in Florence. And we immediately realized that while several of these ligands would induce changes in the chemical shift, which would correspond to binding, to the formation of a complex, protein ligand complex, several others just didn't bind. And irrespective of the Ki and therefore of the affinity constants toward the target. And that was then, at that time, we then did the dose and time dependence, dose response analysis and time response analysis by treating cells with different concentration of ligands or for different incubation times. And then by analyzing the dose dependence and the time dependence, we realized that the reason why some of these compounds would not bind at all in the cell was due to the fact that the diffusion through the plasma membrane is the rate limiting step in the case of these ligands. So what is really useful at this point is a way to overcome the time limitation of in-cell NMR experiments, because what we did for these experiments I showed you, each time we prepared a sample, then we made a cell pellet in a 3mm Shigemi tube, and this is the highest possible cell density you can get in an NMR tube. But it also means that in a few hours the cells will start dying because of hypoxia and starvation, and they will acidify the medium. To overcome this issue and increase the lifetime of the sample, there is the need of a flow bioreactor, which has been developed by several groups. And it has been shown that it can really overcome this problem and allow much longer NMR experiments. That is the main objective of the previous studies, although there is some really nice examples by Alex Shechtman of the real-time NMR applications of a bioreactor. And that is exactly what we wanted in this case. We wanted to monitor these protein ligand interactions in real time in living cells. So to achieve that, we have adapted a commercial device, which is a flow unit that you can just put. It's developed by Bruker, and you can just put it in a NMR spectrometer with a 5mm probe, or even a 3mm probe will work as well. And by introducing a way to keep the cells confined in the flow unit of the inside MR device, then the device itself allows pumping fresh media within the tube. That means that if you constrain the cells in the tube and then you provide fresh media, you can keep the cells alive for a long period of time. So we had developed a few variants, 2 variants mainly, and the second one is the classical one. So I would say the more often used around the world, which is the approach consists in encapsulating the cells in agarose threads. And by doing so, you can confine the cells into the NMR tube and then just flow the medium through the inlet, which goes at the bottom of the tube, and then it diffuses through the cells, which will be here, and then it goes out. And with this approach, we obtain very good cell viabilities estimated by track and blue permeability assay for up to 72 hours. Then we also checked that the metabolic activity was preserved, and that was verified by measuring phosphorus signal from ATP. And as you can see, with the flow, with active flow of nutrients for 3 days, you have a pretty much constant ATP level. Whereas if you interrupt the flow, then you have 0 ATP because it gets consumed immediately. And if flow is switched off immediately, then it also goes to 0 immediately. So that's proof that the metabolic activity is sustained. So by doing so, then we introduced the cells in the tube and then treated the sample by adding the ligand of interest in the reservoir. And that allowed us to make movies of the histidine signals, histidine proton signals from the carbonic anhydrase, which would shift position and move to the bound state. So you can see here, these are just the single 1D spectra of the time series of acetazolamide and metazolamide. Then to analyze in an efficient way this data, I relied on a multivariate curve resolution alternate least square fitting algorithm, CRALS, which is implemented in MATLAB as well as in other programming languages, and it's very straightforward to use. It basically can retrieve the pure spectral components in a mixture as well as the concentration profiles. So that means that from the time series, we could obtain the pure components, which we already know, but it's a very useful way to verify that the algorithm did the good job, because I know what to expect from the spectra. And then we got the binding curves as a function of time, which is what we want. These can be fitted with, for example, these two, acetazolamide and metazolamide, as they diffuse through the plasma membrane, then we assume the very fast binding step when they are inside the cell. So with the diffusion limited binding, we could retrieve the apparent affinity constant and the permeability constants. And these differences in permeability constants are consistent with previous data and also with the in vivo activity of these two drugs. This method can also be applied to 2D spectra, because they can be, let's say, deconstructed and made equivalent to just a very long 1D spectrum, to say it in a very easy way. So the algorithm can also work on 2D spectra and retrieve concentration profiles of two species. So this was shown with superoxide dismutase, which is an enzyme that we have studied since a long time. It harbors two cysteines, which make a physiological intramolecular disulfide bond. So the oxidation of these cysteines can be triggered by treating the cells with external molecules, for example, epsilon, which oxidize these cysteines. And that could be monitored in real time as it happened in the NMR spectrometer. So you can see the oxidized form of the protein going up. So as we can also use 2D NMR, we thought that we could separate better the signals of carbonic anhydrase 2 as it bound different ligands by using 2D spectra, proton nitrogen spectra. So what we did was perform amino acid type selective labeling with the N15 label histidines. And these are the same signals, a subset of signals that you could observe in the emitter region of the CA2 proteins. So they actually belong to the protons, to the histidine protons of the protein. So when we treat, these are in vitro spectra, but when you treat the protein with different ligands, you can you obtain different set of chemical shifts, which means that you can really tell which ligand is bound to the protein, which means you can measure independently each species of the intracellular protein if you could do that in the cell. And it turns out you can. So this is an example of the bioreactor run in which we sequentially treated the cells expressing CA2 with one ligand first, which is the red signal here, and then a second ligand which has a higher affinity with respect to the first one. So the blue is the first is the free protein, and then the red is the one bound to the first ligand, and then the yellow is the one bound to the second ligand. We are now applying this approach to perform competition binding experiments. This is still a work in progress, but I'll show you this as an example result. So the approach consists in treating the cells with the first ligand, which is the reference ligand, so that in order to have 100% fully bound protein to the first ligand. Then at that point, we introduce in a stepwise manner a second ligand, and we keep constant the concentration of the first ligand. So these two are methylamide as reference ligand and SLC0111, which is a drug candidate which is currently in clinical trial as an anti-cancer drug, and it inhibits carbonic anhydrases. So by treating cells with increasing concentrations of SLC, we get a sort of steps. Now it's very noisy, but when taking averages of the steady states, I mean, at each step, at some point we reach a thermodynamic equilibrium where you have a population of protein bound to methylamide and SLC. And so by doing the average, you get the competition binding curves. And in principle, you can obtain ratios of KDs, intracellular KDs by this approach. So I think this is quite encouraging, because you can tell that the first, I mean, each single spectrum is quite noisy, but then the reconstructed spectra are beautiful. They look as good as those obtained in vitro. So I want to thank my team, my supervisor Lucia Banci, and then all the people involved in the CERM in Cell NMR team, and then Claudio Supuran group in Florence, and then Radu, which at the time worked in Strubi for the HEC expression system, and then the Bruker. And thank you all for your attention.